Thomas finished his PhD in Lille in 2017, and he worked with Jamal Khalifa, and he worked on that one was the GEX-15 interactor with protein phosphatase type 1, and then he moved to Karina Rock for his postdoc, where he worked on RNA-binding proteins abundant in apicomplexa, or RAPs. And he's continuing with the RAPs and another organellar gene regulation at Montpellier now. So I'll move over to Thomas to start.
Thank you so much. Hello, everyone. So today I'm going to talk mainly about RNA binding protein and what we call RNA-dependent protein. And in the second part of the talk, I'm going to talk about gene regulation in the organelle of Plasmodium falciparum. It's not working. Ah, it's working. Okay, so as you know, Plasmodium falciparum is by far the most virulent species causing malaria. The parasite has a complex life cycle involving 2 hosts, diverse cell types, and the parasite from various developmental stages. And so the parasite, that means that the parasites evolve in different cellular environment. So the parasite needs to tightly regulate its life cycle while having atypical characteristic. The parasite is unicellular, it's a haploid genome, and it has a limited number of genes, and 1/3 of these genes still have a unknown function. And the last thing, it's the parasite has a regulatory mechanism that differ significantly from what we know in human and model organism. So in gene regulation, RNA binding protein, or RBPs, are really essential at the post-transcriptional and translational level, since they are involved in all of the steps of RNA. For example, they are involved from transcription to the degradation, and it's also includes all of the splicing steps, translation, the localization, and everything. So they are really important for all of the steps of the RNA life. In human and in model organism, thousands of them are already identified. But in Plasmodium, at least I would say a few years ago, there was a limited number of large screen data set concerning RNA binding protein. What we know at the time, it's we had 2 different in silico analyses, and both were based on how to say that in silico analysis based on the well-known and conserved RNA binding domain. And there was only one experimental large-scale capture of RNA binding protein using PolyDT beads that was done in my previous lab. RNA binding protein in Plasmodium, I would say the best example, it's probably the one that you know, it's the DOSI, the ALBA protein, that repress mRNA in female gamete sites. So it's, I would say, the best example and the main RNA binding protein characterized in the parasites. So far, the RNA binding protein are still poorly identified, and as well as they are not characterized in the parasites. So the first part of the project was to identify what we call RNA-dependent protein at the proteome-wide level in the parasites. And this project was done at the University of California in Riverside in the lab of Karin Lorach. So here it's an example of a ribonucleoprotein complex. So by definition, in this complex, you have at least one molecule of RNA, and you have RNA binding protein that interact directly to this RNA. But you can also identify what we call RNA-dependent protein. So RNA-dependent protein bind directly or not directly to this RNA, but the function of this protein is really dependent of the presence of the complex. So if you don't have the complex, this protein, I would say, lose their function. So the function of this RNA-dependent protein is dependent of the presence of RNA. So what we wanted to do is not just to identify RNA binding protein, but identify RNA-dependent protein. So for that, what we did is we use the RDIP method. It's a method based on the sucrose gradient. So briefly, you take your parasites. So here at late stage, asexual stages, you do your protein extraction. And for one part, you do a DNase treatment. It's what we call the control. And for the other part, you do your DNase plus RNase treatment. It's what we call your RNA sample. And you load your protein on a sucrose gradient. Is it good? Okay. And you by ultracentrifugation, you separate your protein. So in your control, for example, if I reuse my protein complex as an example, in the control, you expect your protein to stay in a complex. So you have a complex with high molecular weight. So you will detect your complex in the fraction of high density. However, without RNA, you expect your protein to be disassembled. So either you will have protein alone or smaller subunits of protein. But what you will have is what we call the left shifting of the protein, because your protein has a lower molecular weight than the full complex. And by collecting different fractions, you can analyze where are each protein of the parasites by mass spectrometry or Western blot. So what we decided to do in collaboration with Dr. William Noble at the University of Washington, is to calculate the cumulative distribution function for each protein. So using this method, we were able to generate this kind of profile for each protein. For example, here you have the example of a splicing factor. And as you can see in blue, so for the control, using this method, you can determine that you have almost 80% of the total abundance of the protein in the last fraction, meaning that your protein is in complex and a large complex, we said. However, without RNA, you can see now that we have almost 70% of the total protein abundance detected in the two first fractions, indicating that now your protein is alone and not in complex anymore. And so using this method, we can do some stats. And you can clearly see that we have what I call the left shifting, and indicating that this protein is indeed an RNA-dependent protein. On the right, we have a negative, I would say negative example. So kinetochore protein, and as you can see, the profiles for the control and for the RNAs is exactly the same, indicating that in this case, this protein is not dependent of the RNA. So we use this method for all of the protein detected in our mass spectrometry. And for almost 900 of this protein, we detected the left shifting, suggesting that they are RNA-dependent protein. We did a gene ontology enrichment analysis on this protein, and we observed that the majority were associated with RNA processing, mRNA splicing, and ribosome, confirming that our RDIP method works, and we identify proteins that are associated with RNA. We compare our RDIP result with, I would say, the list of proteins already known to be associated with RNA. And we observe an overlap of 40%. You have to keep in mind that the RDIP doesn't detect only RNA binding protein, but detects proteins that bind also to RNA-binding protein. So that means plenty of this protein were not detected before because they are not RNA-binding protein. And among this specific, I would say, RD protein, we notice the presence of a lot of unknown protein, suggesting that they are probably associated with RNA pathways. But surprisingly, we notice the presence of 14 AP2 transcription factor. This result may suggest that this transcription factor needs to stay in complex in presence of RNA. So the question is it with long coding RNA, or is it due to the fact that they stay in complex if we have active transcription? That's another question, and I don't have the answer yet. So far, what we did is we look at each protein individually, but as the RDIP is based on the presence of the protein in complex or not, we decided not to look at the protein level anymore, but also to check at the complex level. So for example, what we did here, it's 2 examples. So we have the U6 complex. So we check the distribution of all of the components of this complex. And we observe that all of the protein are indeed in the same, detected in the same fraction, suggested that they are still in complex in our RDIP experiment. We did the same, for example, for the 20S proteasome. And you can see that we have a nice correlation for this 2 examples indicated so that all of the components are still in complex. And what we decided to check now is each complex dependent of the presence of RNA or not? So here on the right, you can see it's a distribution, so the cumulative function for all of the components of the U6. And in the RNS condition, you can see that we still have the left shifting compared to the control, confirming that the U6 complex is indeed RNA dependent, which makes sense since the U6 is well known to be associated with non-coding RNA, and it's a complex involved in the splicing. For the proteasome complex, we compare the same both conditions, and we didn't see a statistical significance, indicated that this proteasome is not RNA dependent complex. We did that for more than 40 complexes, and we noticed that in general, complexes involved in RNA pathways are RNA dependent. But we also took advantage of this paper. So in this paper, they how to say that? They use electrophoresis and mass spectrometry combined with machine learning, and they identified almost 500 or 600 clusters of protein-protein interaction. And we also analyze all of these clusters to see if they have a good correlation in our experiment, and also if they are RNA dependent or not. So here, for example, you have the example of the cluster 410. It's composed of 4 proteins, and you can see that we have exactly the same distribution for all of the components, suggesting that indeed this protein seems to be in complex altogether. But what is also interesting is 3 of them are known to be associated with RNA polymerase 1, while 1 of them still have an unknown function. So you can imagine that this protein may be involved also in the RNA polymerase 1. I would say it's guilty by association. And for this cluster, we also notice that this one is RNA-dependent complex. Finally, we also wanted to validate some of the candidates that we identified in our RDIP. So for today, I will present only these candidates. So it was a putative RNA binding protein that was significantly shifted in our RDIP. And we generated a custom antibody against this protein. And we were able to confirm the result obtained by mass spectrometry. So here we can see the left shifting between the blue and the red. And we were able to confirm this by Western blot, validating that our mass spectrometry provide exactly the same result than by a classical Western blot. We took advantage of the fact that we had an efficient antibody, because it's not always the case in Plasmodium, to do immunoprecipitation. And we noticed that this protein was interacting with a lot of protein, a lot of ribosomal protein, but also plenty of splicing factor. So suggesting that indeed this protein may be involved in the splicing function. But our RDIP result suggested that this protein is an RNA-dependent protein. But we didn't know if the protein was really a RNA-dependent protein or a RNA-binding protein. So for that, we use the eclipsing methods. So for those that are not familiar with this technique, it's similar to the GRiP-seq method where you identify DNA-protein interaction. But for the eclipsing, you identify RNA-protein interaction and only direct RNA-protein interaction. So briefly, you do a UV cross-linking, which is specific of RNA-protein interaction. You pull down your RNA-protein complex using antibody. You separate your different complexes by with electrophoresis, and you isolate your regions of interest, and you release your RNA, and you do a classical Illumina sequencing. And for our candidates, what we observe, it's we obtain the significant reads mainly on intergenic regions, so on the 5' and 3' UTRs, and also on the intronic regions, which confirms the results that we obtain with immunofluorescence, suggesting that this protein was interacting with splicing factor. But what was really, I would say, surprising and interesting, it was for the VAR gene. So this candidate was interacted with VAR gene, but not everywhere. It was interacting only on the intron of the VAR genes. So on the bottom here, you have the structure of one VAR gene. So as you know, there is two exons, and in the middle you have one intron, and this intron has a bidirectional promoter leading to the expression of the long-coding RNA sense and a long-coding RNA antisense. And now, if we check in detail our eclipsing result, we notice that the orientation of the read was matching perfectly with the orientation of this, we said, bidirectional promoter. With at the end of the intron, we have the forward reads going so on the plus trends, while at the beginning of the intron, we have the reads going on the minus trends. So maybe, in fact, this candidate was not interacting with the VAR gene's RNA, but maybe this VAR gene also interact with the long-coding RNA. In fact, we'll have to do more experiments to decipher if it's really interaction with the transcripts of with the long-coding RNA or both. But so far, we don't have the data. So just the take-home message I will say for this first part, it's RNA-binding protein and RNA-dependent protein are still largely unexplored in Plasmodium, but not only in Plasmodium, I would say in all of the apicomplex and parasites. And using the RDIP, we identified almost 900 RNA-dependent protein in Plasmodium falciparum, and this technique is good not only to detect this type of protein, but also to check at the complex level. So for example, you can reuse this data to see if your complex that you have in your lab also is well, I would say, well detected in our RD. And you can see if your complex is RNA dependent or not, just by reusing our data. For the second part, which is linked, I will say, with the first one, I will now talk about gene regulation in organelle in Plasmodium falciparum, and I will mainly talk about the atypical RNA-binding protein family called RAP protein, and RAP stands for RNA-binding domain abundant in apicomplexan. So I started this project at the UCR with Karen Lorock, but it's a project that I'm now continuing at the LFI in Montpellier. So the RAP protein are really interesting for many reasons. The first one, it's because they are enriched in apicomplexan parasites. So in 2004, so here you have the first paper describing, identifying this family. And what they notice, it's they detected far more, they predicted far more RAP protein in different Plasmodium species compared to the 6 in human and the 3 or 0 even in yeast. And by prediction, the prediction suggested that this RAP protein are putative RNA binding protein. I reanalyzed this data because they were kind of outdated, I would say. And in fact, I detected far more RAP protein in Plasmodium falciparum, but not only in falciparum. So with 22 predicted RAP protein, but we still have only 6 in human. So which is really interesting for us for, I would say, therapeutic purpose. But what I notice is that these RAP protein are not only abundant in apicomplexan that we thought at the beginning, they are also well abundant in chromerids, I would say, in the alveolata group. And they are also, I would say, highly abundant in the chlorophyta. So the expansion of the RAP protein during the evolution was not just limited to the apicomplexan group, but it's more larger. It probably includes all of the alveolata. The second characteristic of this RAP protein, it's they have a specific structure, what we call the helix-turn-helix structure, with a RAP domain at the C-terminal regions. The location of this RAP domain seems to be important because the vast majority of the RAP protein always have the RAP domain in C-terminal regions. But on the upstream of this RAP domain, we have what we call the helix-turn-helix. And this helix-turn-helix motif, it's what we observe also with the PPR protein, so the pentatricopeptide repeats protein, that are well known and well described to be involved in post-transcriptional regulation in the chloroplast, but also in the mitochondria. So they are well described mainly in Arabidopsis and higher plants. And what we know, it's in fact this helix-turn-helix motif form like a platform to interact with a nucleic acid and in the PPR protein, mainly to interact with RNA. So we can imagine that a similar process, we have a similar process for the RAP protein with this domain interacting and recognizing RNA molecules. For the RAP domain, the structure suggests that the domain can have endonuclease activity, is not I would say confined, because there is some key amino acids that are conserved, some are missing. The limit of this domain is not clear, but it's a small domain of 60 amino acid. And as I mentioned earlier, the C-terminal region seems to be really important for the function. But in general, the RAP domain still have an unknown function, but is well conserved in Apicomplexa, but also if you compare RAP domain with the one in human, they are well conserved. The third characteristic of the RAP protein is they have a specific localization. So either in mitochondria or in plastid. So here you have the 6 RAP protein. They have a different name in human, but it's still RAP protein. And as you can see, they are all described to be in the human mitochondria. They have different functions. So some play a role in the stability of the mRNA, some have more a function of on the ribosomal RNA, but their function is still not well understood. RAP protein were also detected in plants, for example, in Arabidopsis thaliana, inside the chloroplast. So all of this data indicates so that the localization of the RAP protein is really specific. In Plasmodium, as you know, there are two organelles, so the apicoplast and the mitochondrion, and both from endosymbiotic origins, and they are both considered as promising targets. The apicoplast because it's an apicomplexa specific plastid, and the mitochondrion because even if it's, I would say, present in human, it's really divergent compared to the human one. Indeed, if you compare the human and the Plasmodium mitochondrion, they are really, really different. So in human, you have hundreds, thousands of mitochondria per cell. You have only one in the parasites, at least in the sexual stages, I would say. But what is really interesting for us, it's even if they have still oxidative phosphorylation, TCA cycle, all of these machineries and mechanisms are not as well conserved that we can expect, and that means they can be targeted, and it's why, for example, the atovacon is well working against Plasmodium. So in our case, for the RAP protein, we predicted the majority of the RAP protein in the mitochondrion, and only a couple of them, so 3 or 4, that will target the apicoplast. So what you have to keep in mind is all of the RAP protein are encoded from the nucleus. Okay, so they will be imported so by the apicoplast or by the mitochondrion. And the last characteristic I would say it's the RAP protein seems to be essential. So this result was based on the piggyback results. And on the 10 RAP proteins that were predicted at that time, all of them seems to be essential. So based on all of these characteristics, we thought that the RAP protein will be interesting therapeutic targets. So we decided to characterize some of this RAP protein in Plasmodium falciparum. So for today, I will just present 2 mitochondrial candidates and 2 apicoplast candidates, but I will not describe all of our results. So for all of them, we use the same technique. We use the tetradosi system. So we generated the line in collaboration with Dr. Jacqueline Niels from the MIT. And so the tetradosi system is based on the repression of the translation. So you have the binding of the tetradosi complex on the aptamers, and this repress the translation of your gene of interest. However, if you incubate with anido tetracycline or ATC, you repress your repressor, so you have normal translation. So first of all, we wanted to confirm our, we said, prediction. So on the left, you have the RAP21 that was predicted to be in the mitochondria. And as you can see, we have a perfect colocalization in the sexual stages with the mitotic tracker, confirming that the RAP21 is indeed a mitochondrial protein. And on the right, you have the case of the RAP08, which was predicted to be in the apicoplast. And you can see perfect colocalization with the CPN60, and this protein is apicoplast chaperone. So for these two examples, we were able to confirm that we have indeed a RAP protein in the mitochondria and some in the apicoplast of Plasmodium. Next, we wanted to take advantage of our inducible system. So first of all, we wanted to be sure that our knockdown was working. So on the left, you still have the RAP21, and you can see that after 70 hours of incubation without ATC, we have indeed a knockdown of our protein, at least is not detected by Western blot. For the RAP08, the efficiency of the knockdown was even more faster because we were not able to detect the protein after one cycle. So as you can see here on the left. And what we also did, it's, as you can see here, it's this system, it's reversible. So if you add again your ATC, we were able to express again the RAP8 after. So here it was 4 hours, 4 cycles without ATC, but after 6 cycles, we can re-express the protein again. So now, as we know that our system is working, we check if this RAP protein were essential for the parasites. So on the left, you still have the RAP21, so the mitochondrial protein. As you can see, we observe a drastic drop of parasitemia without ATC after starting between 72 hours and 96 hours. And phenotypic analysis confirmed that, in fact, the parasites were fine during the first cycle. So they were parasites were able to reinvade, but they start to be blocked in trough stage, and in fact they die and they never reinvade, confirming so that the RAP21 is indeed essential for the sexual stages. On the right, you have the apicoplast candidates, so the RAP08. We also observe a significant impact on the parasitemia. This one was not completely lethal, so as you can see with the picture on the bottom. But we observed that some parasites were dying, some were still okay, but the protein was essential because if you keep them for a long time, you will have a drop of your parasitemia. But at least RAP08 can be compensated for several cycles. So now we know that these proteins are essential, but we wanted to know what is happening inside the parasites. So for that, we did some RNA-seq. So here is the result for the RAP21. So we took different time points, and what we observe, we observe significant changes after 72 hours, which match perfectly with the first phenotypic changes. And what we observe, it's genes that were upregulated, were genes involved in the RNA processing, translation, RNA processing, ribosome biogenesis, suggesting that indeed the RAP21 is involved in the RNA pathway. And there was really a cool result that we obtained with the RAP21. It's we check the expression of all of the RAP when RAP21 was knocked down. And as you can see here, in fact, almost half of the RAP protein were upregulated suggesting that maybe the parasite try to compensate the lack of RAP21 by overexpressing other RAP protein. And but this mechanism was not sufficient since the parasite were dying. We also noticed that when RAP21 was knocked down, the 3 mitochondrial genes were also impacted. So this RNA-seq data confirms that the RAP21 seems to be associated with RNA metabolism and translation. For the 2 apicoplast candidates, so the RAP03 and 08 on the bottom, what we observe is we observe a lower number of significantly impacted genes, but we observe the detection of apicoplast genes, so genes that are encoded by the apicoplast genome. And we observe that several of them were indeed downregulated when RAP03 or RAP08 were knocked down, suggesting that in fact the RAP03 and the RAP08 have an impact on the apicoplast gene expression, which is matched with their localization in the apicoplast. Similarly to what I presented in the first part, so far we don't know if the RAP protein are real RNA-binding protein or not. So we reuse the ecliptic methods. Here you have the result for the RAP01 and the RAP21. So it's both are mitochondrial candidates. And as you can observe, there was no specific reads on the 14 nuclear chromosome. The same on the apicoplast genome, but we observe specific reads only on the mapping on the mitochondrial genome. And in fact, we observe an enrichment of by 8 times the number of mitochondrial reads compared to the parental line. So that means the line that is not HE tagged. Similarly, for the 2 apicoplast candidates, we observe an enrichment by 30 times reads mapping to the apicoplast. And when we check in detail the results, so here for the RAP01 and the RAP21, so we, for example, here we just focus on the mitochondrial genome. We observe that this specific reads map only on ribosomal RNA that you can see here in green. So the mitochondrial genome is composed of only 3 protein coding genes, and you have also more than 30 ribosomal RNA that are highly fragmented. And it's the ones that you can see here in green. And in fact, you can see that for the RAP01 and the RAP21, these 2 RAP proteins interact specifically and directly only with ribosomal RNA. And you can say that because also you can see that there is a perfect match between the orientation of the reads and the orientation of your ribosomal RNA. So for example, in red for the forward reads, and you can see that it matches perfectly on the ribosomal genes that are on the plus strand. And when you have a blue peak, it's when you have your ribosomal RNA on the minus strands. Similarly, we check for the 2 RAP in the apicoblast, so the RAP03 and the RAP08. We observe for the RAP3 that this one was able to interact with a protein coding gene in the apicoplast genome, but the majority of the reads were mapping here on apicoplast tRNA. So this RAP seems to be really specific of apicoplast tRNA, while for the RAP08, this one was interacting directly to the large, to the gene encoded for the large and only the large apicoplast subunits. So with all of these results, I think I raised more questions that I gave you answer compared to the beginning. So you are probably asking why there is an expansion of RAP protein in apicomplexan. Why these RAP protein are interacting with organellar ribosomal RNA or tRNA. And I don't have the final answer to that. That's why I'm here now in Montpellier to answer to this question. But I have some, how to say that? I have more information that may help you to understand that one. So as I mentioned, the apicoblast is an apicoblast specific plasmid. The apicoblast has its own circular genome, and what you have to know, it's the transcription of this genome is polycistronic. For the mitochondria, when you compare it to human, in the parasites, the genome is linear. It's three times smaller than the human one. In both cases, you have a polycistronic transcription, but you have a huge difference. It's concerning the structure, I would say the composition of the genome. In the human, the mitochondrial genome encodes for 13 protein, 22 tRNA, and only 2 ribosomal RNA here in orange. They are already in full length. So you have one for the large and one for the small subunit. In the parasites, the composition is completely different. So you have only 3 protein coding genes. So here in blue, you have 0 tRNA, and you have 34 ribosomal RNA that are highly fragmented compared to the human one. So what you can imagine, because you have a transcription and polycistronic transcription, it's for the RNA processing of this large polycistronic transcript that you have to cleave, you have to reassemble the ribosomal protein. You need protein for their stability, for everything. So what you can imagine, it's in Plasmodium, this regulation is really important and require far more protein that are probably not conserved in human because you have completely different composition of the genome. And this regulation in human is well studied. So we know that we have more than 1,000 mitochondrial proteins that are imported in the mitochondria to do this RNA processing. So it's plenty of RNA-binding protein, but also you have the involvement of non-coding RNA. And we have what we call the tRNA punctuation model. So here you have, I would say, the human mitochondrial genome, even if it's circular. But we have to keep in mind, it's here in green, you have the position of the tRNA. And as you can see, between 2 mRNA or 2 rRNA, you always have a tRNA. And what is used by the human cell, it's a tRNA punctuation model. So it's the RNA-sp complex plus the RNA-Z that will cleave your tRNA during the maturation. And by cleaving the tRNA leading to the maturation of the tRNA, you will also cleave your mRNA and your rRNA, since you always have a tRNA between 2 different mRNA or rRNA. However, in the parasites, you don't have tRNA. You don't have tRNA, so you cannot reuse the same model to explain the cleavage between all of these different transcripts. Plus, for example, all of the protein of the RNA-sp complex are not well conserved, are not even conserved at all. So all of this model cannot be applied for the parasite. So you can imagine that we have involvement of specific and non-conserved protein, and maybe it's the function of the RAP protein. So we can think about the RAP protein because, as I indicated, they interact with organellar ribosomal RNA and tRNA. Their structure suggested that they may have endonuclease activity. So maybe some of these RAP protein are involved in the cleavage. Some maybe will take care of will have a role of chaperone, take care of the stability of this RNA or tRNA. And this is also confirmed by the publication of the two publications that show that that publish, I would say, the structure of the mitoribosome in Toxoplasma gondii. And in these two papers, what they detected, they detected 5 RAP protein. So here that you have here, that in fact were part of the mitoribosome. So probably, in fact, some of these RAP protein interact with ribosomal RNA. They will stabilize this ribosomal RNA and they will bring them to construct the final ribosomal mitoribosome structure, and they will stay in place to be sure that the ribosome, I would say, the final structure of the ribosomal RNA is still in place. And it's why probably these RAP proteins are staying at the periphery. One other things that suggests that the RAP protein are involved in this gene expression, it's, as I mentioned, there is an expansion of RAP protein in apicomplexa. And what if you check the structure of the mitochondrial genome in this organism, you will see that you have a high fragmentation of the ribosomal RNA. So not only in the apicomplexa, but also in chromerid and in general in alveolata. And you also have, I would say, a fragmentation in chlorophyta, even if this fragmentation is not as important as the ones that we observe in apicomplexa. This suggests that the expansion of RAP protein is associated with the fragmentation of the mitochondria. So because this genome is highly fragmented, this requires more protein to take care of the stability, all of the steps, and it's why you have expansion of RAP protein. So I know that we still have to understand the mechanism, all of the molecular actors involved in all of these steps, and it's why I'm here now in Montpellier. And I hope I will give you all of this answer in the following seminar. So before to go to the question, I would like to thank my former lab at UCR, so especially Dr. Karen Leroc, all of my collaborators, the fundings, and the LFI in Montpellier, where now I will continue to study the gene regulation in the organelle. Thank you so much.
Thomas, that was an excellent talk. Thank you so much. So questions on the floor and also online? Anyone? You could just raise your hands and I could unmute you. So while people are waiting, maybe I could jump in and ask a few questions. So I was quite intrigued by your RD data. I was just wondering, maybe I missed it. Was it from a mixed culture, or were there any time points?
So we use late trough, I would say early chizons, just because you have far more protein at that stage. Okay. So you can probably redo it with tight stages if you want to do it ring, things like that. But you need a lot of material because you have like 25 fraction to analyze by mass spectrometry. So you need to have enough protein in each fraction to detect something. So it was just easier to do it late asexual stage.
And with respect to the RAP data, I mean, obviously most of the RAPs are organelle related. So is there any, I was just wondering, is there any other conserved family of RNA binding proteins that you think would be involved in the nuclear, functioning in the nucleus?
In the nucleus,
I mean, obviously there should, yeah, there should be some post-processing happening in the.
yeah, I guess in the nucleus it will be more conserved to what we have in human, because I don't have the answer. It's probably yes, more or less the same. But what we know also, it's for the organelle, of course. So you have the RAP protein, but we also have the family of the heptatricopeptide. Probably you heard about them. So probably we still have to know what the synergy between them, and also what the difference between the RAP and the HPR protein, because some are classified in both family. So we still don't know really. Yes.
Oh, yeah.
Thank you very much. That was a really nice talk. I was wondering if you've looked at the elution profile of your RAP proteins in your RDeep data to see if you can find potential interactors that maybe have a similar profile. I don't know what kind of interactions they're forming in the asexuals of Plasmodium, but is there any idea of potential interactors there?
So in the RDeep, we observed that 7 RAP proteins were significantly shifted. So at least 7 of them are in RNA-dependent complex. The problem with what you mentioned, it's if you don't know already, you have an idea of what are the components of your complex. It's really difficult just by using the RDeep to detect protein-protein interaction, because you have too many protein in each fraction. So you will have hundreds of proteins that have a similar profile or close profile similar to what you have with your RAP protein. So I'm not sure it's enough to estimate interaction like that. I'm not sure if it's clear. The sensitivity of the resolution of the RDeep is not enough to have a good candidate like that, I would say.
Again, about the RDeep data, obviously your hits are the left shifted ones, but I saw you also had right shifted ones. Is this just noise, or is this a specific protein class?
I checked in detail. There was not like a specific family or class of protein. What we think it's this protein now is not necessarily noise. It's probably protein that were really that interact with proteins that were in complex before. So now that the new protein are available after the RNase treatment, there is probably new interaction. So some protein interact now with proteins that were not free before.
Yes. No more questions. Thanks again, Thomas, for an
Thank
very
you so
interesting
much.
Cheers. Then, yeah. See you later.
Thank you. Have a good day.
Thank you.